ecology stuff at all.
Yeah, forget the ecology stuff. This is what matters.
The ecology stuff is important. Okay, so in as much as we're going to, we're almost through animals, right? So the phyla you need to know, where you need to have a picture in your head, is when I say Porifera, Cnidaria, Ctenophora, Platyhelminthes. We're skipping these. Mollusks, annelids, nematodes. We just finished arthropods. That's all we've got left. The deuterostome. So we've worked our way through all of these protostome animals. Now we're going to focus on the two major deuterostome phyla. There are others, but we're going to focus on the main ones. This is an updated tree. Shows that sponges are paraphyletic. You've all written in that placozoans are the potential sister group to the rest of the Eumetazoa. Aseals used to be platyhelminths. They've been pulled out now as potentially the sister group to the rest of the bilaterally symmetrical animals. Pinafores and Cnidarians are broken apart. This idea of the radiata is probably not real. And then, of course, all of these animals that are bilaterally symmetrical with true mesodermal tissue, we're dividing largely into these bilaterian groups, the deuterostomes and the protostomes. We've already covered the Lophotrochozoan, which includes the major phyla of platyhelminths, annelids, and mollusks, the Ecdysozoans, which includes the major phyla nematodes and arthropods, and now we're going to focus on the deuterostome animals, which include echinoderms and chordates. And I know I said that lightning speed, but I've said it 10 times. You've seen this slide 100 times, right? You all burned that into your brain, right? All right? So here are the deuterostomes. Echinoderms, which I'll definitely get through right now. These are going to be basically what? Sea urchins, starfish, brittle stars, sea cucumbers. These are acorn worms, hemichordates. This is a separate phylum that you're not going to be responsible for. Interestingly enough, there's an acorn worm in a jar upstairs. They ended up putting it on the cart when we were doing annelids. They pulled it off the cart because it's not an annelid, but you'll see it kind of looks a little bit like an annelid. You could imagine it. It's segmented sort of the way that an annelid is, and it looks kind of like a worm. Here we go again. With everything that's called a worm, it has nothing to do with anything else that's called a worm. Here's yet another group of animals that are referred to as worms that have nothing to do with the other worm things. I'll put it on the card so you can see it next week, even though we're not really covering that phylum. What we're going to focus on, aside from the echinoderm deuterostomes, are the chordates. This phylum is huge, and this is the one that most of you will probably find interesting because it's the one that you belong to, right? This includes a few invertebrate chordates like tunicates or ascidians, sea squirts, and all of the vertebrate animals, which means all of the fish, all the amphibians, all the reptiles, all the birds, all the mammals, everything from a minnow to a hummingbird to a blue whale to you is in this group. So this is a massive, huge group. And in fact, I have a separate little PowerPoint I'm frantically going to try to get to on just tetrapods. Tetrapod, 4 limbs, which is going to be basically amphibians, reptiles, birds, and mammals, the guys that have 4 limbs, because that in and of itself is a massive group, yeah. Okay, so huge group of animals. So right now let's go through the echinoderms. So this is going to be the first of the 2 deuterostome phyla that you really have to know. Echinoderm means spiny skin, right? These guys are bilaterally symmetrical, at least as juveniles. These are in the bilateria, but you'll see that as adults they revert to a form of radial symmetry. Pentaradial symmetry. What does penta mean? 5. 5-parted radial symmetry. And you're going to see this across all of the echinoderms. They're triploblastic. That's not a surprise. They're bilaterians. They have mesoderm, right? They have radial cleavage. Now, you all know what that means, right? Nobody's going to confuse that with radial symmetry, right? What does radial cleavage mean again? They have indeterminate cleavage, right? The cell fates are not established on the first division of the zygote. Like us, they can have identical twins, right? You could do a couple rounds of division of the zygote and you could pull a cell off and it can grow into a complete individual. The cell fates are not fixed on that first division of the zygote. This is probably the ancestral condition for all animals, and it's retained in things like cnidarians. It's retained in some of the protostomes. Things like rotifers have radial cleavage. And it's also retained in these deuterostome animals. So you find some radial cleavage across, and then you also have all these different types of determinate cleavage patterns that are derived. So like spiral for platyhelminths, mollusks, and annelids, idiosyncratic for all of the Ecdysozoa, right? So, but those, presumably, the determinate types of cleavage are derived. These guys have an open gut. Again, not a big surprise. They have a mouth and an anus. In this case, of course, since they're deuterostomes, the blastopore becomes the anus, right? And the mouth develops secondarily. And these guys also have a true coelom, but unlike the other coelomate animals that we looked at that have a coelom that arises from a splitting of mesoderm as a schistocele, this coelom arises from outpoppings of the archintron. So it's called an enterocele, right? They have this particular phylum have a spiny skin, and they have an endoskeleton of these little calcareous plates that you'll see when you all dissect your starfish next week. Endoskeletons are unusual for invertebrates. Notice we didn't really have one. You maybe could argue that the fin of a squid is an internal or an endoskeleton, but there's very few animals that we looked at so far that have that. Obviously, vertebrate animals do, right? So it's more of a thing we associate with vertebrate animals. Well, these guys have it too. They have an internal skeleton, one that's underneath their skin. If you asked me, what could I put as a synapomorphy for echinoderms? What would be a character that presumably evolved on the branch leading to echinoderms? Something that is just unique for them, that all of them have and nobody else has. This idea of a water vascular system would probably be it. When you dissect your starfish, you'll see a starfish is anatomically very simple. It does basically three things. It eats, it reproduces, and it moves. That's it. That's basically all a starfish does. The it moves comes from this. How does it move? It moves with a system of water-filled canals that it controls using hydraulic locomotion to operate 2 feet and move itself along. Nothing else has a system like this, with these little ampulla, which are part of the water vascular system, that have little valves that can operate all over the 2 feet. It is a unique type of locomotory system. There is nothing else that has something quite like this. Another interesting thing about echinoderms is that they are all marine. There are no freshwater members, there's no terrestrial members. In most of these phyla, when we're looking at like annelids, there are plenty of marine annelids, but there's freshwater annelids and there's terrestrial ones too. For mollusks, there's plenty of marine ones, plenty of terrestrial ones, plenty of freshwater ones too. Not these guys. These guys are all marine. There's none that have ever gone into freshwater or terrestrial habitat. You're getting good with these larval forms, right? Like when I say sidipid larva, you say ctenophore. When I say planula larva or polyp, you say cnidarian. When I say parenchyma larva, you say sponge. When I say trochophore larva, you say mollusk or annelid or maybe something like that, right? When I say naupliar larva, you say brine shrimp or one of the little crustaceans, maybe what you call barnacle or something like that. Okay, Diplerula, that's going to be your larval stage for echinoderms. There's also a later stage called a Phytoneria, but Diplerula would be a good one to know so that you know a larval stage for an echinoderm. Important to notice that the larvae swim around. The adults, though, are benthic, not sessile, because they move around, right? But they're benthic, they live on the bottom. Because they have really simple body plans, they tend to regenerate really well. You probably know that about starfish, that they can regenerate. Sometimes if you have enough of the oral disc, it can regenerate a whole starfish from an arm and part of the oral disc. They typically have separate sexes and external fertilization, so they shed gametes into the water and sperm the egg fuse somewhere outside in the water. So this is just to remind you on the coelom. This is the type of true coelom that develops in something like an annelid or a mollusk. This is the type of coelom that you'd see developing in a starfish. So from outpoppings of the archintron, you're going to actually see that in some slides that we'll have next week. This shows you the example of a trochophore larva and a Diplerula larva, which would be characteristic of an echinoderm, of an intracoelomate animal. So you just, it looks different, right? They're cilia in different places. It has different little ridges and grooves on it, right? They're different type of larvae. So these, I think, are sea urchins. You're going to look at starfish. So like, I don't know, back in the 80s when I took invertebrate zoology, one of the things we did is they take some sodium chloride and they inject it, or potassium chloride, I think it is, and they inject it into sea urchins. Have you all eaten sea urchin gonads? If you go to like a sushi place, it's called uni or something like that, right? It's bright yellow. And they basically, if you cut open a sea urchin, you pull those out and that's what they eat. But if you inject potassium chloride into them, what they do is, if it's you put them into 2 different containers, right? So you've got a male and a female. The male will shed sperm, the female sheds eggs. And then you can take some of the eggs and you put them under a microscope like this, and then you add a few little sperm to the edge of the cover slip. And you'll see as each sperm fertilizes an egg, as the egg is fertilized, this thing called a fertilization envelope will go boop, like that through the fertilization envelope, and then that prevents any other sperm from getting in. So you can do that for hours of fun, right? You keep adding sperm and you watch the little eggs go boop, boop, right, as they all get fertilized. Then it starts to cleave, so you have a 2-celled stage and a 4-celled stage. Here in this 8-celled stage, which it's hard to get it to look exactly like this, but this kind of shows that idea of radial cleavage, right? In the sense that these 4 lined up on top of the 4 that are underneath them, as opposed to being twisted a little bit, which is presumably what you'd see in something that had like spiral or some form of determinate cleavage. Then you go to a 16-cell stage. Eventually you get to something like a little blastula. And you'll notice at this end it's undergoing gastrulation. So here's the very early formation of the archintron and the blastopore, which in this animal is going to become the anus, right, because it's a deuterostome. Here's the mature archintron, and here you can see these little outpoppings of the archintron, which will ultimately give rise to the coelom. Here are, these aren't Diplorula, I think these are bicunariate larvae, they're a little bit further along, but this I think is all a sea urchin. I have prepared slides for next week in lab that are all starfish. So you can see pretty much the same type of thing, starting with the zygote and looking at these different developmental stages, and you'll be able to see the formation of the intracoel in these guys. Okay, so if this is the phylum Echinodermata, we can divide it up into classes. So there are 5. Here we go with names again, right? Aster, Oidea. Aster means star. Those are the starfish, basically, sea stars. Ophiuroidea are going to be brittle stars and basket stars. And I have those little serpent stars in the tank. I think some of you probably saw them. They call them serpent stars because when you put a little bit of food in there, then they all start to wiggle their arms around. They look like little snakes or something. So I'll show you pictures of those in just a second. Echinoidea are going to be urchins, and also things like sand dollars. Y'all know what a sand dollar is? Right? They sell them in gift shops and stuff. They sell the test, or the endoskeleton, right? And they have that thing on the back that looks like a little marijuana leaf, right? Which shows the what? Pentaradial symmetry. It's 5-parted, right? In Spanish, the name for urchin is erizo. The name for sand dollar is erizo plano. What do you think that means? Flat urchin, right? It's an urchin. And sea biscuit's not the horse, but there's another thing called a sea biscuit that kind of is between. It's like not as flat as a sand dollar, but it's not as round as an urchin either. And I have a sea biscuit up in the lab, so you'll be able to see one, a test of one. So that's Echinoidea. Crinoidea would be things like sea lilies and feather stars. And this group, if you're interested in fossils, you might know. I mentioned briefly ammonites, which is a group of cephalopods that left a very good fossil record. Trilobites is a group of arthropods that left a really good fossil records. And the crinoids within echinoderms left a spectacular fossil record. There just are tons of these. I even have a box upstairs I'll show you next week with a little fossil one in it. At the University of Chicago, they had in the Geosci building, they had this huge plaque that was a whole bunch of crinoids. It was spectacular looking, and that's what I always think of them. I think of the fossils, but there are living ones too, these sea lilies and feather stars. And then the Holothuridae are sea cucumbers, right, which are not cucumbers. Right? If you go into a Chinese restaurant, you order sea cucumber clay pot, you're not eating a cucumber, you're eating an animal that's related to a starfish, basically. And there's lots of these and these and these at most, so you'll see lots of examples of these. It eats, it moves, it reproduces. And this picture is a little unfortunate because it makes it look like every arm is different. That's not true, of course, right? Every arm is the same. You'll see when you're doing the dissection, if you screw up one arm, you just go on to the next one, because they're all exactly the same, right? On the outermost part, when you first open the arm, which you'll probably notice first are these digestive ceci, right? There's 2 stomachs, right? This pyloric stomach and cardiac stomach. We tend not to talk about anterior, posterior, dorsal, ventral with a starfish because it's not bilaterally symmetrical as an adult. Instead, we say that there's an oral side, which is the other side, that's where the mouth is, and then the back side here we would say is the aboral side, but we tend not to want to say dorsal, ventral because that implies that there's an anterior, posterior, which there really isn't. There's no head here, you realize. So it has, the mouth is facing down, this is the anus here, right? And it has these 2 stomachs. Food is brought in through the oral side, right, through the mouth. And then you have digestive juices are produced here. Nutrients are passed down through the arms, through these caloric ducts. That's the eating part. Right next to these, you'll see that there are gonads. And depending on whether your starfish is reproductively active when they killed it, you could have really huge gonads or could have little itty bitty gonads. I've tended to find that the females have larger gonads, but you're not supposed to be able to sex them that way. What we'll do is we'll take a little bit of the gonads, make a smear, put it under the scope, and you'll see it'll either have little teeny tiny sperm that look just like little sperm, or it'll have these huge eggs. And that's the way you're supposed to sex them. But every time I've looked, the big gonads are always on the females, the little gonads tend to always be on the males. So that's how it has sex. Basically from these gonads, it's going to release either sperm or eggs into the water. And then it moves, right? And so remember, it has that water vascular system where it uses hydraulic locomotion to move around. There's a single opening to the water vascular system. It's called the madreporium. It's surrounded by this little thing called a seed plate. You'll see that in your starfish. That connects to a stone canal, to a ring canal, to radial canals. They go to these little ampulla, these little water balloon-like things, each with a little valve at the base that operates the ampulla, or the little podium or tube foot here. And then you have this whole system of ossicles, of these little bony plates that are inside, these calcareous plates that are inside. You'll notice that there's a groove on the underside of each one of the arms that the tube feet come out through. That's called an ambulacral groove. And so these are ambulacral plates. You'll notice that the radial canal comes down through those ambulacral plates. Very simple body plan, right? So, and we'll dissect this. You'll see what it all looks like in real life, but it's a very simple body plan. They have a true coelom that actually forms part of the water vascular system. And sometimes I can get these starfish that have these really beautiful little papulla, which are these little gills, gill-like structures, extensions of the coelom that they extend up through their skin that allows them to breathe or perspire. Okay, so here we go. Asteroidea, this is the picture in your head, right? Everything from something like a nice big helianthus sunstar to just regular little starfish, crown of thorns, picture in a starfish. So next time you walk past a gift shop and it has little dried starfish, you go, oh look, there's some asteroid echinoderms, right? You have an asteroidea and echinodermata. It's going to be what? Ophiuroidea. These are brittle stars. Basket stars. These tend to be able to move a lot very quickly. We'll take one of the little brittle stars upstairs, flip them over, and you'll see that he'll be able to right himself very quickly. They have these sort of long, slender arms, and the ossicles are not tightly interlocked so that they're able to move much more freely than most of these other, all of these other groups actually, that may be criner. Okay, these are echinoderms that are in the class Echinoidea. So these are the tests of an urchin, like this little Strongylus introtus. That would be a purple urchin like you find in California.
What is it?
Strongylus introtus is a genus for that. Notice I can remember that. 20 years I've taken invertebrate zoology. At one point I used to know all of the algae and the invertebrates in California really well. You could walk me to any tide pool, I could name every single one. But I remember this. This is Strongylus introtus purpuratus, because he's purple. The purple sea urchin that you find in California.
I remember Oncorhynchus meerkat from my grade.
Yeah. They stick with you. And this right here, of course, is a live sand dollar. And you can see very well here, it's pentaradially symmetrical, right? They all have this underlying pentaradial symmetry. If you look carefully at the test, you'll see always 5. And even something like a sea cucumber, if you look end on, you see 5. Okay, so that's Echinoidea. These are Crinoidea. So like I said, most of them I think of as fossils, but there are living members, these sea lilies or sea feather stars, they call them sometimes. And then these are Holothuridae. This is a sea cucumber. Very often at most they'll have these chocolate chip sea cucumbers. Some of them are these tiny little guys, and then some are like that big one that's me on the Great Barrier Reef with a big sea cucumber. One of the most interesting things to me about these guys is that they are capable, if they're being attacked or bothered by some kind of a predator, of taking all of their internal organs and they can actually spit them out through their anus and then just grow new ones later on. So they just kind of like let whatever's bothering them eat all of their insides after they've spit them out through their anus, and then they just grow new ones on the inside. just walk away like nothing. Wouldn't that be kind of cool if you had something chasing you like a bear or something, you could just sort of spit your liver and all of your guts and everything out through your anus and go, oh, that's cool, I'll just go do it. That's pretty amazing when you think about it. That's so disgusting. Okay, so what would a good synapomorphy for echinoderm? See, didn't I tell you back in January you weren't going to escape this phylogenetic terminology? Here we are. Hopefully you all know what a synapomorphy is, do you? What is a synapomorphy? It's a character that evolves on the branch leading to the last common ancestor of a clade or a monophyletic group. Something that can define that group, right? So I might point out, if you look at echinoderms, they have radial cleavage, radial indeterminate cleavage, right? They have an intraceal, a trucelome, completely lined with mesoderm that derives from outpockets of the archenteron. They have a water vascular system with all these little canals that control operation of tube feet and allow for hydraulic locomotion, right? And they also have an open gut, right, with a mouth and an anus. So they have all of those, right? You're trying to find the one that defines them, the one that presumably is evolved on the branch leading to the last common ancestor that all echinoderms share. The other three characters are likely what type of characters? Not synapomorphies, but simplesiomorphies. They're things that evolved further back in the tree and would be true for other groups too, not just for echinoderms, so they can't be used to define them. See how I told you this was going to keep coming back and back? You didn't really believe me to come back all this much, right? But here it is. Here we're getting right to the end, and I'm still doing it. Because if you look in the performance standards for this class, you'll see that that's exactly what it says. It says something about understanding the changes in taxonomy and why the changes are occurring. And a lot of it has to do with interpretation of characters, right, when you're inferring phylogeny. Okay, good. So water vascular system is good. All the other things there would be true for chordates as well, right? So those are not going to be things that you could use to define echinoderms. Okay, how about a sand dollars in the same echinoderm class that contains food? Starfish, basket stars, sea cucumbers, or urchins? Okay, one more. Is there one more? And I'm going to get up. I'm ready to start on chordate here. Maybe about 15 minutes ahead of you guys. Okay, here we go. Urchins, right? Erisoplano. How about this one? The radial symmetry in echinoderms has a common origin with the radial symmetry found in cnidarians. Do you know who cnidarians are? That's one about the 9 phyla you're supposed to know. Sea anemones, jellyfish, hydra, ophelia, right? Those are all radially symmetrical. So is the radial symmetry that you find in echinoderms, does it have a common origin with the radial symmetry that you find in cnidarians? Or does the radial symmetry in echinoderms represent a derived condition, in other words, something new for them? Or is it primarily found just in the larval forms of echinoderms? Or are none of those correct? So what can you say about the radial symmetry that you find in echinoderms? You are all focused on radial symmetry, right, and not radial cleavage. Right? You really do know the difference between those. You realize those have absolutely nothing to do with one another at all. Okay, is everybody there? One more? Is there one more? Okay, here we go. Yeah, that's pretty bad. So like 3 of you out of 15 got it right. It probably represents something new for them. Where are echinoderms? What major group of animals are they nested up inside of? The bilaterians. They're bilaterians, right? And in fact, their larval forms are bilaterally symmetrical. It's only as adults that they're radially symmetrical. Very unlikely that that could have a common origin with the radial symmetry that you see in cnidarians that are extremely distantly related, that are completely outside of the bilaterians. The most likely scenario is that it represents a derived condition just for echinoderms. Yeah. So do the 12 of you that just missed that, do you know why now? Yeah, no, maybe. It has nothing to do with radial symmetry in cnidarians. Here we are, last animal phylum. So what do you have to know? Porifera, Cnidaria, Ctenoptera, Platyhelminthes, Mollusca, Annelida, Nematoda, Arthropoda, Echinodermata, and Chordata. Those are the phyla you have to know. You don't have to know brachiopods or bryozoans or rotifers or any of the other ecdysozoans like onychophorans or tardigrades or anything. I mean, it'd be nice to know all those, but those are the ones you really have to know, right? Porifera, Cnidaria, Ctenoptera, Platyhelminthes, Mollusca, Annelida, Nematoda, Arthropoda, Echinodermata, and Chordata. You'll notice the first little part of Chordata here. Bilaterally symmetrical, triploblastic, radial cleavage, which means indeterminate cleavage, open gut, deuterostome, meaning the blastospore becomes the anus, and they have an intracoelom. True coelom that develops from outpopping to the architron. You'll notice that that looks exactly like echinoderms, right? You'll notice that they have segmented muscles. Segmentation involved multiple times, right? We had segmentation in annelids, we had segmentation in arthropods. That was part of the reason people thought they went together. You're segmented. How are you segmented? Is there a little part of your body that appears in series or segments like this? Your vertebral column. Your ribs, right? You're segmented, right? It's one of the characteristics of chordates. Chordates have segmented muscles. They have jointed skeletons typically. If you have a jointed skeleton, you have to have muscles that work in antagonistic pairs to bend and straighten joints. A muscle that bends a joint is called a flexor. A muscle that straightens a joint is called an extensor. So just like arthropods that have jointed skeletons and have flexors and extensors, you have a jointed skeleton, so you have flexors and extensors. There are exceptions to this. I mean, like for example, if you look at a snake or something like that, it has intercostal muscles that function as longitudinal muscles to allow it to undulate, right? So there, and fish could also have muscles that are arranged that way. But if you have, especially if you have appendages the way the tetrapods do, then you're going to have these flexor and extensor muscles. They're mostly separate sexes. Hagfish have hermaphroditic members, but for the most part, they're separate sexes. But what really makes a chordate are these four things here. So you're going to like really get this, right? A notochord, nerve cord, gill slits, and a tail. Notochord, nerve cord, gill slits, and a tail. Notochord, nerve cord, gill slits, and a tail, right? Those are the four characters that define chordates. Every chordate has these four characters at some point during their life. That's an important thing. At some point during their life. So you don't have to necessarily retain them into adulthood, right? In fact, like there's a couple of those that you don't, right? So a notochord is a thin, flexible rod that runs along the dorsal side of the animal. In a vertebrate animal, the notochord becomes the backbone, right, or the vertebral column, right? You also have a dorsal nerve cord that runs along the notochord. In a vertebrate animal, that becomes your spinal cord, right? So your spinal cord, your vertebral column. You'll see in the case of something like a sea squirt or tunicate, right, that they lose the notochord and the nerve cord as an adult. They don't have them at all, right? They're invertebrate animals, but they are chordates because they have a notochord and a nerve cord. As juveniles, they have these characters. Pharyngeal gills, where's your pharynx? It's here, it's in your throat, right? So pharyngeal gill slits is very different than anything we looked at, like gills in a mollusk, right? Like the ctenidium, or gills in your crayfish, right? Or gills in a nudibranch or something, right? This is a very different type of gill. These are throat gills, and you're going to go, well, gee, I don't have throat gills. You did. Yeah, you did. The idea is you don't have to retain them into adulthood, right? Obviously you have lungs now, because if you had gills in your throat like a fish does, They wouldn't do you much good if you're a terrestrial animal. So you lost these, but during development, right, when you were embryonic, you had gill pouches, you had pharyngeal gill pouches. And a muscular post-anal tail, you might have noticed that of the animals we've looked at so far, nothing really had a tail, did it? Not a real tail, not a muscular tail, certainly. Chordates do. And again, you're probably going to say, well, I don't have a tail. Well, you did when you were very, very small. And of course, some babies are even born with tails. Usually they take them off. Most parents don't like their kids running around with that tail. But every once in a while, you even have little human babies that are born with little tails, right? So you definitely did have one at some point. Yeah, so you're all going to get that, right? Notochord, nerve cord, gill slits, and a tail. Every chordate has that at some point during development. So if you ask me what the last common ancestor of all chordates look like, I would guess it looks something like this. Right? It's a very simplistic little cartoon. Shows a one-way digestive tract, right? Has a mouth and an anus. Obviously, this was the blastophore. The mouth developed secondarily as these little segmented muscles here. But it has this notochord, this nerve cord, that would become, in a vertebrate chordate, the vertebral column, the spinal cord, right? It has a muscular post-anal tail, and it has gill slits in its pharynx. Has the 4 basic characters that a chordate would have. Nothing that I've shown you up till now, no other animal group has this, right? No other animal group. In fact, most animal groups that I showed you so far, if they had nerve cords, they were where? They were ventral, right? With a dorsal circulatory system. This is almost like, and most of the invertebrate animals we've looked at flipped upside down. It's going to have a ventral circulatory system, although it's not considered a hallmark chordate feature. But this idea of a dorsal nervous system or a dorsal nerve cord, that's brand new, right? I haven't mentioned that for any other group, right? That's not going to be found really in any of the invertebrate groups. It's only going to be found within these guys, within these chordates. Okay, so now is when this starts to get really bad. If we were doing this 10, 15 years ago, when I did it with my high school students back in the 90s, I would have said phylum Chordata, and then I would have taught you these as classes. Urochordates, which include the sea squirts. Cephalochordates, which are lancelets. I'm going to show you a picture of it in a minute. Agnathan, do you know what naph means? G-N-A-T-H, like the little gnatho bases on the horseshoe crab. Those are his little things he chews with, so it means jaw, right? So gnatho means jaw. So agnathan would be jawless fish, things like lampreys and hagfish. Chondrichthyans, ichthy means fish. So chondrichthyes, these are the cartilaginous fish. Who would those be? Sharks, stingrays, skates, right? The osteichthyes, who are those? The bony fish, right? That's going to be basically every fish you could possibly name. If I said name 100 fish, don't say shark, skate, ray, hagfish, or lamprey. You're going to name 100 bony fish. Every fish you could name. Perch, salmon, trout, goldfish, whatever you say, it's going to be a bony fish, right? And these were divided into 2 groups. The actinopterygians, which are the ray-finned bony fish, right? That have little bony rays that come out through their fins, but they don't have skeletal elements that extend beyond the body wall. And the sarcopterygians, which would include everything from like a coelacanth, do you know what a coelacanth is? To lungfish that have little bony elements that actually extend into their fins, almost like little arms and legs, right? And then amphibians, reptiles, birds, and right down there is mammals. Unfortunately, this class right here is not monophyletic. You all know what this means now, right? This group right here is not monophyletic unless you add all of these guys in here. I'm going to show you the tree in a second. This is why I was saying on day 1, you're a bony fish, because I can't make bony fish a real thing unless I add all of the tetrapods. It's a paraphyletic group without tetrapods. Now you know what that means, right? Y'all know the problem we've got here, right? Reptiles is paraphyletic unless you add birds. So I now have what historically was a class, and there's another class inside of it. That's a mess. And Sarcopterygia is also not monophyletic unless you add all of the tetrapods.
Is there by any chance that they possibly could be monophyletic? I just haven't figured
Of course
it out
it could.
yet.
Of course it could. In fact, I shouldn't tell you things like this, but I just saw something that there are some people that are challenging now the whole idea of ecdysozoa. This group that I just taught you like it was something that I got from God and presented to you as truth, it's now being challenged. After just having put it into all the textbooks and everybody rallying around it and everything, there now are some people that are challenging the whole idea of ecdysozoa and whether or not it's actually a monophyletic group. Yeah, anything I tell you, you realize is the thinking of the moment, right? You realize in 10 years, if you took this class, it'd be totally different. The classification, the taxonomy would be. But the good news is in 10 years, if you took this class, you know the organisms at least. So now you could go, oh, wow, you know, now they actually think paramecium is related to a flamingo or something, or whatever, right, that they're onto at that point. At least you'll know what a paramecium is, and you'll know what a flamingo is, so that you can start putting it together some new way. You realize the classification I'm teaching you is radically different from what I learned 20 years ago. But that's just the nature of this, you realize. It's always going to be changing as they gather new evidence and do new analyses. So, yeah, everything you're learning right now. But notice that's why we talked at the beginning about the nature of science. I mean, if you're not comfortable with what you're learning being potentially wrong, you realize nobody would have ever learned science ever. I mean, what they were taught 100 years ago in some little schoolhouse on the prairie somewhere is probably all wrong, you know what I mean? But what do you say? Well, they shouldn't have gone to school at all. They were just wasting their time. I mean, you kind of have to do the best you can with what you got at the time. You guys happen to, hopefully what you're learning is closer to actual truth than what they learned 200 years ago. You got to wonder. But hopefully what we think now is a little bit better, but it's undoubtedly not completely right. It's going to be different in 10 years. That's just the nature of this. That's the way science works. Science is always changing. So you'd never be able to study anything if you waited for it to be perfect. Okay, so here we've got all the deuterostomes. There's the echinoderms, and here are all of the chordates. You'll see some of those, but like these, Myxini and Petromyzontida, those would have been agnathans. Can you see how that can't be right? We can't have that. These three would have been the Ostictians, the bony fish. That's not monophyletic, unless you add all these in here, right? What would you call everybody from Myxini down to Dipnoid, do you know? Starts with an F.
Fish.
Fish. Word fish doesn't mean anything, unless you're a fish. Because then I could include all the way down to here, right?
Unless you go fishing.
Unless you go fishing, right? Fish just means it has fins and it lives in the water. That's all fish means. It doesn't mean anything from a relatedness or from a genuine evolutionary group point of view. So here's where we go. So those old class names like Agnatha are gone. Names like Sarcopterygia are gone. Ostictians now is going to be used to refer to this whole group. Notice we want all of these different names to be applied. We want to be able to talk about the entire phylum Chordata, right? But we also like to be able to talk about these guys. A head evolved right here. Nobody prior to this has a head. So we'd like to be able to talk about everybody who has a head, and we can give them a name. We can call them cranians. You'll see that the only ones that are vertebrates that have a vertebral column are these guys. So maybe we could call this monophyletic clade vertebrata. How about these guys? They all have jaws. We could call them gnathostomes, jawed mouth. If we use this word Osticthes, it's kind of weird to have ichthy in there, which means fish, but with the osteo part, these are the only guys with an ossified or a bony skeleton. We could talk about the lobe fins that have body elements or skeletal elements that extend beyond the body wall. We could talk about this group of tetrapods that have 4 limbs, and amniotes that have a true land egg. Here's the problem. This is where I'm going to lead you. This is a phylum, right? So what's this then? Subphylum, sub-subphylum, sub-sub-sub-subphylum, sub-sub-sub-sub-sub-subphylum, super-super-super-super-superclass, super-superclass, superclass, and then I guess these can be the classes. The Linnaean ranks don't work very well here. Historically, this was the phylum, and these, more or less, were the classes. Now, all of a sudden, we want to be able to give names to all of these different nested monophyletic groups. There aren't enough ranks. So most people, when they start talking about chordates, they ignore Linnaeus. So we say phylum Chordata, and then we say, oh, you know what? This is a vertebrate, it's a gnathostome, it's an Ostictian, and it's an Actinistian. And they stop worrying about whether it's a class or a phylum. So I told you I was going to do this, right? When we were doing arthropods and mollusks and things where it was convenient to use Linnaean ranks, I used them. And now I'm gonna abandon them again because they don't work very well in here. There's too many newly recognized nested groups. And you also have this problem that a lot of the old classes like Agnatha are not real. You have this problem of having birds inside of reptiles, so the 27 bird orders have to somehow get consumed inside this class called Reptilia that already had 16 orders of its own. So now what's the group birds? Is that an order? Or is it a subclass? Or what on earth is that? It's a mess, right? So I'm going to completely slip away from Linnaeus. Okay, you see how there's going to be way more names than what we've already had? This is what we're going to start with. Don't forget any of this. Don't forget any of this by next Tuesday, right?